Okay, ready to talk about smell. So, remembering back to last week when we first began our discussion on sensations, we learned that there were kind of two major groups of sensations, the generals and the specials. Okay, general sensations, there's actually two types of those. Okay, somatics and viscerals. Somatic included things like, hot and cold, pain, proprioception, um, touch and itch and tickle and stuff that we talked about on Thursday. We aren't really going to cover visceral sensations at this point in our semester. We're going to move on from the general senses to the special senses, running through those. And there are 5 special sensations. The first is smell. Fancy term for that is olfaction. Like all the special senses, has a low threshold, which means that it's easy to create a nerve signal. It doesn't require a lot of molecules, as it states there in the second sentence. Only a few molecules of certain substances need to be present in air to be perceived as an odor. Obviously, the more molecules, the stronger the smell will be, but it doesn't require a lot. We all know that we use our nose for the sense of smell. Within our nose are olfactory sense organs that consist of epithelial support cells and specialized olfactory receptor neurons in the most superior portion of our nasal cavity, an area that we call the olfactory epithelium. Okay, the olfactory epithelium, I'll bounce back before I finish talking, is this kind of generalized purple area, the top of our nasal cavity. This is a look at, looks like the left half of the nose. This would be the lateral wall of your left nose, or left nasal cavity. This purple area is the olfactory epithelium, where you have the actual neurons for the sense of smell. Let's talk about those neurons. These are called olfactory receptors, and they are bipolar neurons, extremely sensitive and easily fatigued. So they're bipolar, meaning that they have the cell body in the middle of the two extensions. Remember, we talked about bipolar neurons in the first unit on nervous tissue. We learned that bipolar neurons were pretty rare. Not a lot of places that we have them. In our nose is one of those places. We can describe them as the first order neurons of the olfactory pathway. They're the first ones that are used in that labeled line that carries the signal eventually up into our brain. At the tip of each of those olfactory receptor cells are what we call olfactory hairs, the olfactory cilium, okay, which, if I actually jump ahead, are located on the dendrite of the cell. Okay, so they basically are just little extensions of our receptor cell, and this is where you find, and this is what this illustration is showing you, a series of receptors that are where we have odorant molecules, the things that we smell, bind, and thus create what's called an action potential. So we'll get into all that here in just a second. So these olfactory hairs cilium are located on the olfactory receptor neurons that touch the olfactory epithelium lining the upper surface of our nasal cavity. These are the receptors that are stimulated by odorants, and they are just tiny hairs projecting from the dendrites of the olfactory receptors. And if you look at this illustration, we show, and it's kind of hard, I know, because it's not the biggest picture, but they're kind of blue shaded cells are the olfactory receptor cells. We've also got some cells in there that are kind of tannish in color. Those would be what we call supporting cells. And then supporting cells do exactly what it sounds like. They provide support and nourishment, electrical insulation, detoxifying chemicals that they come into contact with for those olfactory receptor cells. There's also basal stem cells that replace those olfactory receptors by mitosis, which is cell division. And as everybody is aware, within our nose, we have snot. We don't really call it snot, we call it mucus. That mucus, some of which is produced by what we call olfactory Bowman's glands. And this is very intertwined with the olfactory epithelium. because the mucus helps moisten the surface and helps with dissolving odorants so that they can come into contact, that is, the odorants, the chemicals, with the olfactory hairs. And now, on each side of our nose, there are about 40 bundles of the slender unmyelinated axons of olfactory receptor cells extending through olfactory foramen, okay, which are tiny little holes at the top of our nasal cavity within what's called the cribriform plate, which is from the ethmoid bone. For those of you who are anatomy should remember, that's going to be the roof of your nasal cavity. You can see there are little tiny holes, and these things that look like little strings are the axons of those receptor cells. So when we stimulate the neuron, it then creates an action potential that propagates up and out of the nasal cavity into what's called our olfactory bulbs. Okay, the olfactory bulbs are paired masses of gray matter at the terminal end of our right and left cranial nerves. Cranial nerve number one is our olfactory cranial nerves, as we've mentioned, and you just hopefully knew that for your exam. Inside the olfactory bulbs, which we show right here, one of those olfactory bulbs, we see a synapse occurring between those olfactory receptor cells and the second cells in the pathway. Okay, we call these olfactory bulb neurons, okay, which are part of the olfactory pathway. They then project that signal posteriorly, forming the olfactory tract, as you see here. Some of the axons project to the primary olfactory area in the temporal lobes to allow for conscious awareness of smell. Other axons of olfactory tract the olfactory tract project to the limbic system and hypothalamus to allow for emotional and memory evoked responses to odors. So thinking back to when we talked about the brain, we learned that the limbic system is our emotional brain. Okay, when we smell stuff, we can have emotional responses to it. Okay, smell something that triggers a great memory or a great just a normal wonderful feeling when you smell it. Like, oh, it smells. It reminds me of being a kid again, or something like that. Okay. So smells aren't just something that we smell and say, oh, that smells like whatever. We can also have these more intricate responses as a result, with emotions and memories involved as well. Now, transduction with regard to smell is a pretty complicated little process. Remember, transduction refers to converting stimulus energy into electrical energy, action potentials. Okay. In transduction for smell, we involve, and I'm going to type it up. So maybe there's word. This involves what's called a second messenger system. Now we'll see a second messenger system situation in our endocrine lecture, and I'll explain what a second messenger system is. I wanted to type it so you could write it. Okay, so second messenger system tells you that you're going to have two messengers. The first messenger is an odorant molecule binding to a receptor protein is found on the plasma membrane of our olfactory hair or cilia, as it's labeled here, which then I just always think of it like, a domino effect. If you ever play with dominoes as a kid, you line them up, you push one over, what happens to the rest of the dominoes? They all fall over, right? It's a cascade of events. Okay, the binding of that odorant to that receptor protein activates a G protein, which then activates an enzyme, adenylate cyclase, or adenylin, same thing, adenylate cyclase, which converts ATP 2-cyclic AMP, which is our second of the two messengers. All these steps are not super important, I promise you. But look what happens as a result of cyclic AMP becoming activated. We have a channel that is opened up, and it's a cation channel, so it allows things like sodium and calcium in. And as we know, these are neurons. They have a resting membrane potential, -70. If sodium or calcium come in with their positive charge, what's going to happen inside that neuron? It's going to become excited, and it might hit threshold. And if it hits threshold, we've now created an action potential. An action potential that's then going to propagate through the olfactory nerves to the olfactory bulbs along the olfactory tract, and then to the primary olfactory area of the cerebral cortex. As far as the exam goes, you're not going to have to know all these steps. You'll want to know that this involves a second messenger system, like I was just typing up for you. Okay. And that's pretty much kind of one of the biggest things to take away. Now, moving on from smell to taste. Gestation is the fancy word for taste. Is actually very similar to the sense of smell. It is a chemical sensation. We smell odorants. We taste what we call tastants. So there's odorants for smell, tastants for taste. Okay. Involves structures within our tongue predominantly. There's also what we call taste buds in other places, but the most prominent place we find our taste buds are within what we call lingual papillae or papillae. I've heard it pronounced that way. Little bumps basically on our tongue where our taste buds are located. Okay, I'll talk about taste buds here more in a second. Let's talk about these lingual papillae. Okay, the little bumps on top of our tongue. There's 4 types. Filiform are tiny spikes without taste buds, so they actually have no role with taste. So, they have no taste buds. They're not going to allow us to taste. But they do contain tactile receptors, so they're sensitive to like touch and the texture of food when our tongue comes into contact with it. They also increase friction between our tongue and food to help move that food through the oral cavity. Anybody ever felt like a cat lick you or felt the tongue of a cat before? What's it feel like? Sandpaper. Cats have a lot of this on their tongue, a lot of these on their tongue. The second are foliate papillae, small and on the lateral margins of the tongue. Most of them degenerate in early childhood, so not super important, which leaves us with these 2 for taste. Fungiform, located at the tips and sides of the tongue, contain about 5 taste buds each, but the majority of our taste buds are found right in this kind of inverted V, which happens to be a very handy thing that it is a V, inverted V, because we call these valate papillae, and they're at the rear of the tongue and contain up to half of the taste buds of our entire tongue. Each of them houses about 100 to 300 taste buds. So a very significant amount. Now, it used to be believed, and I remember when I started teaching this like 10 years ago, they used to say that, you like taste sweet things on like on the tip of the tongue, bitter things on different parts of the tongue. That is not true. I don't know why that was ever taught, but it was, as far as not that long ago, something that was still taught. All right, let's talk about taste buds. We can see one zoomed in here in our illustration. Pretty much a very similar situation to what we saw with smell. We have taste for what we call gustatory receptor cells. You can see those are the yellow ones. Banana-shaped have apical microvilli, so they're like the little tiny hairs that we just talked about for smell. These are the surfaces that are going to act as the receptors for the food molecules. Now, these guys are an example of a separate cell. If I go back several slides towards the very beginning of the lecture, We talked about separate cells and how they are not neurons, but they synapse with neurons. And that picture that was just there on that page was one of these same cells. They synapse right here with the first order taste neuron that then carries the signal into our brain, okay, forming the first part of the gustatory pathway. We've also got in purple here basal cells, which are stem cells found at the periphery of the taste bud, and they produce these type of cells, the ones that are kind of dark red colored, the supporting cells, which also contain microvilli, and they actually will then transform into these, becoming receptor cells. So it's kind of a nice life cycle. These purple ones become the red ones, which then become the yellow ones. Because we have to continually replace these receptor cells because they only have a lifespan of about 10 days. Which explains why, you know, you like severely burn your tongue, like when you eat something super hot, like, you know, pizza bites or something like that, which are delicious, but impossible to wait until they're not 1,000,000 degrees. You're not going to lose your taste completely forever. It'll be back without too long. Wait.
So since cats have those like tiny spikes, do they taste a little less than?
I don't know. That's a good question. I imagine they still have the other type as well. And there's a big thing that I was just getting ready to talk about. There's a big interaction between smell and taste. It's like why, if you get a cold, or like right now with people, they say that when they get COVID, they lose their sense of smell. They also lose their sense of taste too, because and you can, you know, trick your. I used to do this when I had a physiology lab. I would get like different flavors of bubble gum, and I'd have students like close your eyes and have a student like a lab partner, you know, take a Big Red, for example, and put it under their nose, and then have them chew on wintergreen, and see if they could tell the difference or tell what they were chewing on. And it was pretty fun to see. Some students couldn't figure out what they were chewing while they were smelling something else. So there's a big relationship there between the two. And like smell, taste is going to detect or require detection of molecules. Once again, we call these tastants. And another thing that I used to do in lab was demonstrate how important saliva is for the sense of taste. If you like take a towel and dry off the surface of your tongue, or if you like hold your mouth open for a minute or two and let the tongue dry out a little bit. We used to do this where we would take sugar and sprinkle it on a person's tongue and let it just sit there with a dry tongue. The sugar doesn't dissolve very well. And it would be like a minute or two a lot of times before a student could actually taste that sugar because it wasn't dissolved. What the saliva does, it helps dissolve that chemical to get it into the little depressions within our taste buds so that it can stimulate those microvilli. Okay. There are 5 primary taste sensations. Salty is the first. It's produced by metal ions like sodium Na+ and potassium K+. Sweet is produced by many organic compounds, particularly sugars. Sour is a sensation usually associated with acids, so things that have hydrogen ions, H+. Bitter is produced by alkaloids. Alkaloids, fancy word to describe your bases instead of acids, which bitter is typically not something a person enjoys tasting. And it causes, you know, animals and us as well to reject the food, which could be in like case of being spoiled or diseased. And then a fun word to say is umami. Actually, I should say unami, I believe, not umami, but that's okay. And meaty taste produced by amino acids, I believe. It's a Japanese word. But now, I imagine everybody's thinking to themselves, you know, I don't just taste sour, I don't just taste sweet. You know, you can taste like combinations of those things, whether it's a orange, vanilla, Coca-Cola, or something else. You know, there's combinations when it comes to, you know, things like flavor and stuff. Okay, let's talk about transduction. So converting and creating action potentials that are then propagated into the brain. Once again, importance is we have to have saliva to dissolve that chemical, the tastant. Once it's dissolved in saliva, it can make contact with the plasma membrane of our gustatory microvilli, which are these little finger-like extensions in our pictures at the bottom of the page. Okay. Once again, we are going to stimulate these cells, creating what's called a receptor potential, and that stimulates the exocytosis of synaptic vesicles, releasing the neurotransmitters that then activate the actual first-order taste neurons. There are 3 cranial nerves that are involved with the sense of taste. So 3 pairs, it really should say, not 3, because there's 6 total, because they're in pairs. Cranial nerves, number 10, the vagus nerve, the glossopharyngeals, those are 9, and was that VII, so that's 7, the facial nerves. And we show in this illustration just one of each, because we're only showing you one side, one tongue, one half of the tongue, I should say, not one tongue, one half of the tongue. So it really should say 3 pairs of cranial nerves. So that's kind of unique. I would definitely make sure you know that those are the 3 pairs of nerves involved with taste. From there, the signal then travels into the medulla and then into the limbic system, hypothalamus or thalamus. Okay, taste signals that project into the limbic system. Once again, our limbic system is our emotional brain, and that can trigger emotional responses. You know, we all know what taste can do. You know, there's emotions that come with it, as well as memories, just like what I was talking about with smell. Taste signals that go to the thalamus. Remember, the thalamus is our gateway to the cerebral cortex, allow for synapsing to occur, where then the signal travels to the parietal lobes to give rise to conscious perception of taste. Now, what has to happen first is exciting these taste receptor cells, and it is going to occur one of 2 different ways. Okay, when we taste something salty, which means it's probably going to have either sodium or potassium ions in it. Remember, sodium Na+ can travel in through sodium channels, and if we taste something salty, sodium is going to rush right into that cell, and that excites it. Same thing for things that are sour with hydrogen ions. We have hydrogen ion channels, so it's kind of a super simple process to get this cell depolarized. Whereas with things that are sweet, bitter, or umami, what we do is, it's just like with the sense of smell, a second messenger system. Lots of proteins are involved, very domino effect, eventually causing a excited, depolarized cell, which triggers the release of those neurotransmitters. Pretty complicated, and for that reason, I'm not going to really require you to focus too much on that. Okay, moving on to our 3rd, and I'll really even say 4th as well, sensations, because we're going to talk about the ear, because our ear is home to receptors for both hearing and for equilibrium. We'll talk more about hearing first. Fancy word for hearing is audition. There's actually 3 parts to the human ear, each of which plays a specific role in the sound waves being converted into action potentials. Okay. The first part of our ear is what we see, what we feel, what we can stick our finger in. It includes the auricle, which is this fancy word for your external ear, as well as the external auditory canal. Then the whole function of that is to transmit sound waves that are created by noises, like the words that I'm speaking right now, to the middle ear. Between our middle ear and the external ear is a fancy structure called the tympanic membrane, which is just a fancy way to describe our eardrum. Whenever a sound wave hits it, think of it like a drum, and that it rattles. Okay, it vibrates. Okay, attached to the eardrum are 3 what we call auditory ossicles. Fancy way to describe our three ear bones, which we just talked about in anatomy, malleus, incus, and stapes. When the eardrum vibrates, the malleus vibrates, which causes the incus to vibrate, which causes the stapes to vibrate. So it is a chain of events occurring where we then transmit that vibration to an opening to this big structure here, what we call the cochlea, which looks like a snail shell. Inside the cochlea are the receptors for the actual sense of hearing. Okay. What we do is we take the energy that strikes our eardrum and transmit it to something much smaller, what we call the round window. Okay. As it states there, the tympanic membrane or eardrum covers the opening to the middle ear. It has 18 times the area of the oval window. So it basically causes that energy to go to a very fine position, specific spot. That energy I'm talking about is these vibrations. Okay, so we're concentrating that vibration to a much smaller area. Now it doesn't show it in this illustration, but attached to some of these ear bones are some muscles, and the ossicles and those muscles help to actually at times decrease the transfer of energy to the inner ear. We call this reflex the tympanic reflex. What they essentially do, the muscles will contract to kind of decrease the amount of vibration that is transmitted inward to prevent like kind of overload into the cochlea. Unfortunately, the reflex has a latency or delayed reaction of about 40 milliseconds, which is not quick enough to protect your ear from sudden artificial noises, stuff like a gunshot. You know, that's why your ears are going to ring and might hurt as a result. Okay. So we're transmitting that energy into the cochlea, which is one of the structures involved with the sensations in our ear. Cochlea is responsible for hearing. These, what we call semicircular canals in this area, what we call the vestibule, are more responsible for sensations of equilibrium or balance. We're not quite ready to talk about balance yet, so we'll stick with hearing. I know it looks like it's cut off there at the top. It's supposed to be hair cells right there. So receptor cells for hearing are known as hair cells. They have the capability to transduce. Remember, transduce means to convert mechanical vibrations into electrical signals. They are located within a structure of our cochlea, what's called the organocorty or spiral structure, okay, and specifically found in one of the three ducts of the cochlea called the cochlear ducts. At the tip of each of these hair cells are about 40 to 80 stereocilia, which are just little tiny hair-like, finger-like microvilli, which are the actual receptors for the sense of hearing. Okay. Here's what they look like. The little microvilli extending off are shown with this. These are also examples, these hair cells are examples of separate cells. So they're not neurons, but they synapse with the first order neurons that carry the signal into our brain, okay? So these hair cells synapse with the first order sensory neurons, and they form what's called the cochlear branch of our vestibulocochlear cranial nerves. If I go back to this picture, looking at the vestibulocochlear nerve that's labeled, you can see there's two branches. One that goes to the cochlea, that's the cochlear branch. The one that goes over here to what's called the vestibule is the vestibular branch. Vestibular branch carries signals for balance, because the sensory cells for balance are located over here. The cochlear branch carries the signals for hearing, because that's where the hair cells are for hearing. Okay. Now, those neurons carry the signal up into our brain stem, synapsing in our medulla with what we call cochlear nuclei. Some of the cochlear nuclei decussate, which means they cross over. We talked about that previously, and they terminate in the midbrain. Others don't, as you can see, so it's kind of crazy. Some signals come over, some don't. Crossover, not come over, crossover. If they don't, they terminate in the pons on each side of the brain. Now, regardless, both signals coming from the pons in the midbrain are then carried up to the thalamus, and the final signal is then propagated to the primary auditory area of our cortex and our temporal lobes. Now, like I told you guys with smell and taste, you're going to have to know all these steps, where it's going to go, what's, main thing is knowing which cranial nerves are involved. That's a big thing. Knowing that, we're going to process that stuff we hear in our primary auditory area, which you guys should already know, because you just had a test over that stuff today. So, to kind of summarize how this all begins, we first have to have the sound waves reach our auricle. Auricle is just a fancy word for our external ear. And then that set of sound waves is going to transfer their way in through the external auditory canal, where they then strike our tympanic membrane, causing the malleus, incus, and the stapes to vibrate. This causes the membrane, the oval window, to vibrate, which is that once again the opening to our cochlea. Inside our cochlea, I haven't talked about this, are two important fluids, perilymph and endolymph. Now, I always use an analogy related to a movie here to talk about this. It was a movie that came out when I was a kid, Jurassic Park. So if you haven't seen it, there's a very important, prominent scene in the movie where they're stuck inside a vehicle, and there's a cup of water sitting in the cup holder, and off in the distance, T-Rex is coming towards them. They don't hear T-Rex, but they see the cup of water starting to rattle, and they see the little ripples occurring in the cup of water. That vibration that I was talking about that strikes our eardrum is then transmitted to our cochlea, causing these fluids to have little ripples in them, essentially. And as the perilymph in the body labyrinth of the cochlea and the endolymph in the membranous labyrinth of the cochlea move, this causes a chain reaction that causes those hair cells to become stimulated, which then results in impulses traveling along our cochlear branch of the vestibulocochlear cranial nerves. So, if you've seen the movie, great. If you haven't, hopefully you understand my analogy there. Now, we can hear different frequencies as far as sound goes, like a high pitch versus a low pitch. If you're a singer, you know what I'm talking about. Baritone versus soprano, that kind of stuff. How do we tell those differences? Well, higher frequency waves are going to stimulate hair cells closer to what we call the round window, whereas lower frequency waves are going to stimulate hair cells near the beginning of the cochlea. And so we stimulate different areas of our little snail shell, the cochlea, and then those signals are transmitted to different areas. You can see yellow, red, and purple are color-coded within our primary auditory area of the cerebral cortex. And so we carry it to a different area of the cortex, and our brain is therefore able to interpret and understand that you might be dealing with a more medium frequency sound or a higher frequency, higher pitch type situation. So to kind of reiterate, what we're showing here is the eardrum, 3 bones. This is a simplified illustration of the cochlea. Sound waves reach the eardrum, it rattles, it rattles, it rattles. That energy is transferred in here. We have the ripple effect occurring, which is going to stimulate the organ of Corti, which is where those hair cells are located. They bend, causing them to create a receptive potential, resulting in a nerve impulse. Not that you're going to have to know all these steps or anything like that. I just wanted to talk about it, really to kind of show you how amazing and complicated hearing really is. Okay. Now, I already showed you the illustration here on this page to show the hair cells and their little microvilli extensions. Notice that the microvilli are not uniform in length. In fact, it's kind of a cascade where it goes from larger all the way down to smaller. Okay. Attached to each of these microvilli are little tip-link proteins that are very cool, and I'll explain what's going to go on here in just a second. Okay, so reading through, hair cells transduce mechanical vibrations into electrical signals. A tip-link protein, which looks like a little slinky, connects each of the stereocilia to one another. And at the tip of each of those is a mechanically gated ion channel, which is the stuff shown here in green. When that fluid ripples, look what happens to those microvilli. They bend either towards the biggest or the smallest finger-like extension. If the bend occurs towards the taller cilia, stereocilia, those proteins open up those ion channels, allowing ions in. That means the cell becomes excited and neurotransmitters are released. And that means action potentials are created in our first order auditory neurons. If the bending occurs in the opposite direction, those channels get closed, which means ions aren't going to flow in, which means we're going to decrease frequency of nerve impulse transmission in those first order neurons. So it's kind of crazy, but it's also super cool at the same time. Now, the great thing about this, it's pretty similar to what we see with equilibrium, which is a fancy way, once again, to describe balance, the ability to maintain coordination, balance, and orientation in a three-dimensional space. Receptors for equilibrium, see the same type of cells. Hair cells, they're just found in a different place in our ear, what we call the semicircular canals or ducts, and two chambers, anterior saccule, posterior utricle. There's two major types of equilibrium, static and dynamic. Static, think stationary. What do I mean when I say stationary? When you're not moving, okay? So when our body is laying still, orienting our head, when our body is stationary. In this situation, the macula saccula lies vertically on the wall of the saccule, and the macula utriculi lies horizontally on the floor of the utricle. If we change our head position, like we turn our head to the side, it produces a change of pressure, stimulating those hair cells that in turn stimulate receptors of our vestibular nerves. I'll show you this all stuff on the next page in just a second. Dynamic equilibrium is the perception of motion or acceleration, which can be linear, angular, so like straight ahead or like spinning, which linear acceleration is perceived by the macula, angular is perceived by the crista. Transduction of equilibrium involves stereocilia located on the surface of the hair cells, depolarized and hyperpolarized in the same way that we just saw with sound transduction. So it's basically the same thing. We have the bending of the cells and stuff like that, which then synapse with the first order neurons in the vestibular branch of the vestibular nerve. So I've included a couple of illustrations here to show you equilibrium and how this all works. Okay, so it is a little bit different than smell, or excuse me, than hearing, because we don't have a fluid that ripples. Instead, we have a membrane that jiggles. Weird. I'm using these kind of non-science terms, but that's okay. I'm just trying to make it sound somewhat, to where make it make sense. So I like to think of this, what we call otolithic membrane, like a Jello mold. Okay, everybody's seen and had Jello before. You know that you take that pan of Jello, and if you shake it, what happens to the Jello? Jiggles, right? Okay. Well, this otolithic membrane is where you find those hair cells with their stereocilia. Okay. And if, for example, your head is nice and straight, the Jello, the membrane, is nice and normal. But if you tip your pan of Jello over, the Jello is going to slide, right? Just like what happens with our head when we tilt it forward. Force of gravity causes that membrane to move. What's going to happen to those little finger-like extensions? They're going to bend. So it's the same process as what we just saw for hearing as it is balance, which is a little bit different, causing the bending. Okay. Regardless, those hair cells for balance are not the neurons, but they become excited, where they then synapse with the first order neurons, which collectively form the vestibular branch of our vestibulocochlear nerves, which carry the signal, as you can see, into our brain stem. So the axons of those sensory neurons in the vestibular branch synapse with sensory neurons in the vestibular nuclei in both the pons and medulla. Now, with balance, we also have a lot of input coming from our eyes, because vision helps a lot with balance as well, which explains why, I had you guys all stand up and say stand on one foot. It's a lot easier to do it with your eyes open than it is with your eyes closed. And that's an easy way to demonstrate the importance in our eyes with balance. We also have information coming in from proprioceptors, which are found in our joints and our muscles and our tendons, or like positions of those structures that play a big role in balance as well. But those vestibular nuclei then integrate the information, sending out signals that control movements of our eyes, help control head and neck movements, and control muscle tone in order to maintain equilibrium. So it's very complicated for, a sense that we really kind of take for granted. I'd say this is the sensation that we take the most for granted, because it's not like, smell and taste how, when we smell something great, it triggers something, a good feeling in our body, or when we taste something, how great that is, or when we, we see things, we hear things. We use equilibrium all the time. The only time we really appreciate it is when we are thrown off of balance, like when we get dizzy, or if you have equilibrium issues, you can get nauseous and that kind of stuff. So other neurons carry signals to the thalamus, as you can see, from the vestibular nuclei, from the thalamus, which once again is our gateway to the cerebral cortex. We then carry the signal to our parietal lobes to provide conscious awareness of the position and movements of our head and limbs. So conscious perception of our balance. Okay, progressively getting a little bit more complex to finally now our most complex of the sensations, in my opinion anyway, vision. And it is 1:30. So you guys can write this stuff out, and we'll just stop here for the day. If you guys want to just finish it, it'll take us like 5 or 10 minutes. You guys have to anyway. Let's just finish it so we can be done with it. How's that sound? Okay, vision is obviously the act of seeing. Pretty amazing are our eyeballs, even though they're very small. More than half of the sensor receptors in our human body are located within our small eyeballs. A very large part of our cerebral cortex is devoted to processing what we see. It's also a very important thing. The retina is a layer of our eyeballs where the receptor cells for vision are located. We call these photoreceptors, and they absorb, or they're the cells that absorb light and generate a chemical or electrical signal. The two photoreceptors are called rod cells and cone cells, or rods and cones, what they're a lot of times just called. They're called rods and cones because of what their tip looks like, either a cone shape or a rod shape. Okay. Rod cells are responsible for night vision. I would definitely know that. Night vision is involved with rod cells. In other words, they cannot distinguish colors from each other because at nights when it's black outside, you can't tell if you're, seeing somebody wearing an orange shirt or a yellow shirt or whatever. Each retina has about 120 million rods. Cone cells are responsible for functioning in bright lights and are responsible for our day vision as a result, as well as color, which you all want to know that. We don't have nearly as many cones in our retina, only about 6 million per eyeball. Rods and cones are not the only cells in our retina that aid in vision. We also have non-receptor retinal cells, which include bipolar cells, which provide a large amount of what we call convergence and stimulate ganglion cells. Bipolar cells are the ones that are stimulated by the rods and cones. Ganglion cells, definitely wanted you to know that they form the optic nerve, which is the cranial nerve that exits our eyeball and carries the signal to our brain. These are the ones that receive signals from the I shouldn't say ganglion cells. Obviously, ganglion cells don't receive signals from ganglion cells. Ganglion cells, as you can see here, are going to receive signals from bipolar cells. So fix that, cross out ganglion and write in bipolar. We also have horizontal and amacrine cells that form connections between other cells and enhance perceptions of contrast, edges of objects, and changes in light intensity. Okay, looking at this illustration, we see those different cells that I was just mentioning found throughout the retina. The retina actually can be broken down into several layers. The photoreceptor cell layer is where the rods and cones are. First thing I want to kind of point out is the number of rods and cones compared to the eventual ganglion cells. I said that ganglion cells form the optic nerve, so that's where the signal leaves the eyeballs. Okay. Bipolar cells, I said, provide for a lot of convergence. Convergence refers to the fact that we go from a bunch to very few. Basically, we end up decreasing. There's a bunch of these, there's a lot less of these, and eventually even less of these. So we don't have to have as many neurons stimulated, which helps with, a lot of things, but perceptions of contrast and depth perception and that kind of stuff play a big role in it. Now, you can see hopefully why we call them bipolar cells. Bipolar cells are bipolar neurons where the cell body's in the middle. We can see rods and cones are structured a lot like that as well, where they then synapse either with those bipolar cells, but you can also see horizontal cells. Hopefully you can see why we call them horizontal cells. There's amacrine cells kind of there in the middle as well. And then eventually the ganglion cells receive the signal, and then there would be the optic nerve coming out of the back of the eye. Now in anatomy, for those of you who are taking anatomy, we'll learn all about the other layers of the eye, which includes the sclera and the choroid. We'll talk about the lens and all that stuff. I won't get into really much about that in this class. On top of the fact that vision is super, super complicated because you have to factor in like changing light intensities and how, if I were to flip the lights on now with it being kind of dark in here, how your vision would be affected, how when we perceive stuff, do you guys remember the blue and black dress or the white and gold dress? Some people saw it as blue and black, some people saw it white and gold. A lot of that was your brain's interpretation of what you were seeing. So I'm going to keep it pretty dang simple. Not even going to get into like creating the signal. I won't talk about transduction at all. Really the kind of last thing I want to talk about is just the pathway for vision. Another really super complicated thing about our vision is that each eye can be broken down into kind of two halves. A nasal half, which is closer to your nose, temporal half, which would be closer, to your temporal bone over by your ears. Okay. Any light that strikes the nasal half, so you can see, will be over here, or the temporal half. You can see signals are going to go to different parts of the brain, meaning that, if you close your eyes and you look with just one eye, what you're seeing throughout your visual receptive field, half of that information is going to one part of the brain, half of it's going to the other side of the brain. So your right eye is going to send signals to your right cerebrum and your left cerebrum. And that's because of what's called the optic chiasm, which is a point where those optic nerves crisscross. Chiasm actually means cross. So that's kind of cool. And regardless, the ultimate destination of what we see goes to the primary visual areas we've seen before, talked about before, which is an area found in our occipital lobes, the back of our brain. I always remember that because, the phrase, I have eyes in the back of my head. We don't obviously have eyes in the back of our head, but we process what we see in the back of our brain in our occipital lobes.
Hearing that reminds me of the story where somebody got like shoved over. They fell and they hit like the back of their head and they